Now speaking about pollination in case of Cycas, pollination is anemophilous in the sense the pollen grains are to be carried by wind, air current. The cells of the here, this picture, the cells of the nuclear beak present in, this is the pollen chamber, this will be the pollen chamber, and this is the archegonial chamber. The cells present in this nuclear beak of the pollen chamber, they are going to disintegrate and lead to formation of a viscous fluid. This viscous fluid is cohesive in nature, sticky in nature, and this fluid is going to ooze out and settle down here at the micropyle end. So the nuclear beak cells present in the pollen chamber, this is the ovule, right? So the nuclear beak cell present in the pollen chamber, they are going to disintegrate and form a viscous fluid. This fluid is going to ooze out, and that is, it is going to come out and settle down over here at the micropyle end. Why? Because it is sticky in nature, and when the pollen grains, they are released, right, at the 3-celled stage, they are going to be carried by the wind, and the wind, when they are going to be settled down on this part or come in contact with this part, here we have the fluid which is sticky in nature. That fluid is going to hold these pollen grains and drag them inside. So this is the procedure for catching the pollen grains and getting them inside the pollen chamber. So we have this fluid which is going to ooze out of the micropyle and collect and whatever the fluid is collected here, and this is called as pollination drop. It looks somewhat like this. So this is the ovule. We have this archegonial chamber followed pollen chamber followed by archegonial chamber. The cells present in the pollen chamber, they are going to disintegrate and ooze out at this point. This is a pollen grain. This enlarged structure is pollen grain. This is pollen drop. Inside the pollen drop, we are going to